Hello, I'm John Scahill. I'm Biological Secretary of the Royal Society. And it's my great pleasure this evening to introduce the 2016 Croonian Lecture. The lecture is named for one of the original members of the Society, Dr. Coen, who, before he died in 1684, decided to propose that there should be two lectures, one given annually, one given here, and the other at the Royal College of Physicians. I don't know whether that one still happens, but this one is the premier biological lecture of the Society. It's a pleasure tonight that our lecturer, Enrico Coen, is going to give us his lecture, as you've seen in the title. He is a molecular geneticist whose career in plant developmental biology has largely been at the John Innes Institute in Norwich. His research addresses how gene expression is controlled during plant development and how the structures of plant components, such as leaves and petals, are generated. He's used theoretical and experimental approaches to these questions and has brought his interests as a talented artist to his research and to the wide communication of his results. We look forward, Enrico, very much to your lecture tonight on Picasso, Pottery and Plants, Hidden Rules Governing the Development of Natural Forms. Enrico.
Well, thank you very much for that kind introduction. I feel totally honored to get this award, and it's wonderful to have our work, the work of our group, recognized in this way. I'd like to begin with a sort of experiment. So here you see 11 drawings done by Pablo Picasso when he was 64. And what I'd like you to do is to choose one of these drawings, the drawing that you would say is the hardest to draw. Okay, so if you could take a second to just choose one of these that you would think is the hardest to draw. Okay, so now if you put your hands up, who chose a bull on the top row? If you could put your hands up. Okay, that's quite a lot of you, that's about a third. Who chose a bull on the second row, on the middle row? Well, that's most of the rest of you. Who chose a bull on the bottom row? Oh, it's about a third. It's interesting. Who chose the last bull, the very last bull? Okay, interesting. So yeah, that's about 10%. Now, in a sense, there isn't a right or wrong answer to this question because it all depends on what you mean by hard to draw. And the bulls on the top row are hard to draw because there's a lot going on. There's a lot of lines and features to capture, as there are also in the second row. But the bull on the last bull, the one on the bottom right, is hard for almost the opposite reason, because you have to decide what to leave out. Okay? And you could say that it took Picasso 64 years to figure out what not to draw, to arrive at what you might call the essence of what he considered to be the essence of a bull in a few lines. And we encounter the same issue in science. So to what extent is science all about giving us the most complete and full description of the world with all its complexity? Or is it about stripping away that complexity to arrive at some underlying simplifying principles? And these two aspects are not unrelated, because in order to arrive at simplicity, often, initially, you need to dwell on the complexity and see how, through that, you can arrive at some underlying simplifying ideas. And what I'm going to do today is tell you sort of some of the ways that I've tried to grapple with this issue as applied to a particular problem to get at the essence of the principles that underlie that problem. And the problem I'm going to be talking about is not drawing bulls, but the issue of shape, the generation of shape. So all these shapes that you see are produced through tissues of cells, sheets of cells deforming into particular shapes, like a flower, or a brain, or a heart, or a butterfly wing. So all these shapes are generated through this process. And unlike Picasso drawing his bulls, there's no external hand guiding that process. It all has to happen through the internal processes within the tissue itself. So somehow the tissue has to miraculously turn itself into these different structures. The problem is how does it do that? So where do you begin to answer a question like that, a problem like that? Well, how did Picasso become good at drawing bulls? So here's a clue. This is a drawing done by Picasso at the age of 10. So he was already drawing bulls at that age, and he continued to draw bulls for the rest of his life. It was a theme that recurred all the time in his working life. And I think it's fair to say that Picasso was totally obsessed with bulls. Why he was obsessed with bulls, I don't know, but the fact is he was. So as a matter of interest, how many of you would say you have or have had an obsession with something? So would you put your hands up as to how many people would say they have an obsession? More hands are slowly going up as you come out. It's a coming out list. So that's a lot of obsession in this room. So what I would say to you is treasure your obsessions, because obsessions can be a great source of joy and inspiration. And I'm going to tell you a bit about my obsession. It's not bulls, it's this flower. Okay, it's a snapdragon flower, which has a rather complex shape. And face view, you can see it has these upper petals that are distinct from its lower petals. And when you look at it from the side, you see the petals are joined to form a sort of tube. And this structure, this complex structure, is organized in relation to its pollination mechanism, so that a bee, in order to pollinate the flower, has to land on the lower petals, open the flower, collect the nectar, and in so doing, it carries out the pollination. So how did I become obsessed with this flower? Well, I wasn't obsessed at the age of 10 with this. I didn't think about snapdragons then. I wasn't obsessed with it at university or during my PhD. My PhD was on Drosophila, on fruit fly genetics. My obsession started a bit later when, as a postdoc, I applied for a job at the John Innes Centre. And then I met this chap, Brian Harrison, who was retiring from the John Innes. That was why the job had come up. And he'd spent a large part of his life working on these snapdragons, but particularly the variegated snapdragons. And he was working on them because he suspected that this particular type of mutant snapdragon was caused by a piece of DNA that can move around the genome from one position to another. That's rather unusual. Most genes are fixed in their positions, but this piece of DNA can move around, and it's called a transposon because it has this ability. So he was fascinated by this rather unusual phenomenon of this possible transposon, and he worked for many years on this together with his research assistant, Rosemary Carpenter, and together they built up a massive collection of genetic material in relation to this behavior of this transposon. And just before I met them, they'd had a very successful collaboration with Heinz Seidler in Cologne, and he'd been able to isolate this transposon and show that indeed it was a piece of DNA that moved around. So that was very exciting. But I was also interested in this for another reason, not only because the transposon was intrinsically interesting, but I knew that maybe we could use transposons as well to identify and study genes that controlled the shape and development of the flower. So as I was walking around the glasshouse with Brian and Rosemary, and they were showing me the emporium of Antirrhinum flower color mutations, I said to them, do you by any chance have a mutation that changes the shape or structure of the flower? And they said, yeah, we do actually, we do. Come over here, come over here. So we went to a corner of the glasshouse and they showed me this. So this rather sort of not particularly spectacular flower they called koi, because unlike the wild type flower, which has these rather showy petals, this flower has has these green sepals or small green structures instead of the petals. And that's why they called it koi, because they thought it was rather modest, not having these showy petals, but these sepals in their place. But when I looked at that flower, I didn't see a modest, retiring flower. What I saw was a fruit fly. So why did I see a fruit fly? For this reason. Because having worked on fruit fly genetics, I knew of these mutations that had an effect on switching the type of structures they make. So the top you can see a wild-type fly only has 2 wings. But below that you see a fly with 4 wings. And that's because that's a mutant in which 2 structures that lie just behind the wings, called balancing organs, have switched their fate so that they now become an extra pair of wings. And that's exactly what I was seeing, or so it seemed to me, with my snapdragons. I was seeing one structure that then switched into another. Instead of petals, we'd seen a switch into sepals. And so by studying these genes, maybe we could understand how these switches operate, how one structure switches to form another. So I was hooked. I thought, yes, I have to work on this fantastic system to try and unravel this problem. And fortunately, they offered me a position. So I joined Rosemary, who stayed on after Brian retired. And for the following years, we started to systematically look and screen for mutations that affect the structure of the flower. So here's one of our fields. In 1989, we'd stroll through these fields. 99.9% of these flowers would be normal. So you'd be walking along, but occasionally you'd get very excited because you'd see a plant with some strange flowers on it, all right? A mutant flower, and that was a gene maybe that was affecting the development of the flower. So we started studying these genes and we started to categorize this collection of mutants, and some of them are shown here. Just as, in addition to the mutant, the koi-like mutant that I showed you earlier, we obtained other mutants. For example, the one on the top right, which doesn't have petals, but has sort of male structures, stamens in place of the petals, another type of switch. Or the one on the bottom right, which instead of having sex organs inside the flower, stamens and carpels, it has a profusion of petals or sepals. So in all these cases, we're ending up with one type of structure switching into another as you go from the outside towards the center of the flower. But there were other mutants as well, mutants that changed the symmetry and shape of the flower. Okay, so here's an example. Here's a mutation in which the flower is now radially symmetric. Okay, and it's actually a mutation in 2 genes called Psych and Dick. When you take away these genes or mutate them, you end up with a radially symmetrical flower shown on the right. And these petals are now all the same all the way around the flower, and they resemble the lowest petal of the wild type or normal flower. Now, because we generated these mutations through these transposons, through these mobile pieces of DNA, we could identify and isolate the genes involved. And then we could see where these genes were active. And they were active from a very early stage of development. And I want to show you that by looking at the expression or the activity of the Psych gene. So if you look at a very early flower bud, you don't see any organs forming. What you see is what looks like a tiny loaf of bread. It's about 1/10 of a millimeter across. It's a tiny little thing. You may be able to see the cells on its surface. This is going to form eventually the flower. But at this stage, there's no obvious asymmetry from top to bottom. The loaf of bread looks rather symmetrical. But if you slice through that loaf of bread, and then you ask where is the gene, the Psych gene, active, what you see is this. You see that the gene is only active in part of the loaf, and the upper part, that's shown in the dark blue. So where you're seeing dark blue color is showing us where our gene is active, and it's active in the dorsal or upper region of this tiny bud. So already, at a very early stage in its development, the flower is already carved up into different regions that are going to form the different parts of the flower, in this case the upper as opposed to the lower parts of the flower. So based on experiments like this, we came up with the following type of model for how the flower is formed. Okay, so we start off with the flower bud as being carved up into these different regions of gene activity. One set of genes is involved in establishing differences from top to bottom, the dorso-ventral pattern, and I just showed you an example, for example, that was expressed in the upper or blue region of the flower, and you have different regions going from top to bottom. And then you have another set of genes that are involved in specifying the types of organs as you go from the outside towards the center of the flower. Those were the other mutants that I showed you that changed the types of organs, sepals and petals and so on, and that's a radial system of identities. And then if you combine these two together, you end up with a patchwork of different regions, each with a particular combination of gene activities. And so each organ in the flower, developing flower, has its own combination, and that determines the type of structure that you generate. So maybe this then was our Picasso's simplified bull. Maybe this was the essence of the flower. It was this patchwork of gene activities that accounted for the different structures it made. But when you think about it, you see that actually that description, although reduced, is missing a lot. And what it's missing is the fact that the flower has some amazing structures and folds, like the shape of the petals, for example. This is a flower which has been sliced through in half, and you can see that beautiful shape and structure of its petals. So how is that related to this patchwork of gene activity? It's entirely unclear how a patchwork like that connects with a shape, a complicated shape of that kind. So that's the problem we then really had to address. Over the next 15, 20 years, we started to tackle this problem of how you generate a shape. So where do we begin? Well, one way of beginning is to start to describe the different stages in development of the flower. So this shows you, this spiral shows you the different stages of the flower bud as it grows and matures. And you'll see one of the key things you'll see is that it gets bigger as you go. It starts off about 1/10 of a millimeter across and gets bigger and bigger through growth. And the amount of growth that's happening is actually much greater than what you're seeing here, because I'm changing the scale bar as we go through development. So if I showed you those all to the same scale, you wouldn't be able to see the earliest stage because it would be so small. So all this shape is being generated somehow through growth. Okay, the question is how? How does growth generate something like that shape? Well, a clue to that problem came from going back to our variegated flower. All right? This, remember, was caused by a transposon, this mobile piece of DNA, and it had these spots or stripes on the flower. Now, to explain a bit more what's going on in this flower, I'm going to just show you one of the petals, the lowest petal, and I'm going to cut it off and enlarge it. So here's now the petal, and what you see are these patches of red on the petal. Now, to see what's going on here, imagine that the petal bud is starting off really small with all its cells are destined or programmed to become white. But as the petal grows, occasionally there'll be a switch, a cell will switch so that it will now gain the potential to make red pigment. And as it grows and divides, that cell will give all its descendants the same properties. So you end up with a patch of cells in which now red can be made, the red pigment can be made. And you can see that if I enlarge one of those patches, I hope you can see the individual cells that make up that patch. So those are all the descendants of a single cell. And each of those patches represents a different set of descendants. Now, when you look at that patch, one of the first things that you see is it's not round. Okay? It's elongated along one direction, preferentially compared to the other. And that tells you that growth is not happening equally in all directions. It's oriented in one direction, preferentially. And moreover, if you look at the pattern of those orientations, you'll see they seem to be coordinated over the flower petal. All right? So there seems to be some coordination of orientations of growth that's involved in the generation of this petal. But it's entirely unclear how these orientations relate to the shape of the petal. That's a hard problem to solve. And to illustrate how that's a tricky problem to get your head around, I went to a pottery friend of mine, Jonathan Keep, and I asked him to illustrate an equivalent problem with pots. And so what I asked him to do is to take two different colored clays, combine them. You'll see he makes a sort of hamburger. He's here in the audience listening with interest, I can see. So he made a sort of hamburger of these two different colors, and then he threw a pot. And what I'd like you to do is, as you see the movie, is try and anticipate and predict what's going to happen to these two different colors as the pot is made. Okay. So here's the hamburger. Okay, and I actually have it here. Here it is. I think it's rather beautiful, actually. But I don't know how many of you anticipated that you would get it's actually a double helix, this. It has a double spiral of red running through the grey clay. So I certainly didn't predict that. And it shows you how difficult it is. Even when you think you know how a pot is made, it's actually quite difficult to predict how the regions are going to deform during the process of its making. And we had almost the opposite problem. We had a pattern of stripes and spots on our flour, and we needed to figure out what was this process that would give rise to that pattern. So that's why it's a tricky problem. So when people are faced with a tricky problem, scientists, one of the things they do is they try to model it and simplify it and try and arrive at some sort of model that would allow them to understand it better. And modeling has a very strong tradition in the biological sciences. Perhaps the most famous example of the use of modeling is this one. Another double helix, discovered or formulated by Watson and Crick. And in arriving at their structure, they were totally dependent on the use of models. What they did was they made cardboard cutouts of molecules, trying to stick them together in various ways, and others with rods and so forth. And so they constructed their model of the double helix, using this modeling, playing with these different structures. But modeling wasn't sufficient to solve this structure. Okay. In fact, their first structure was a triple helix. The first solution that they came up with, they thought DNA was a triple helix and all the phosphates were in the center. That was their initial model. And they actually thought they'd cracked the DNA structure at this point. They were very proud of their model, but they thought they'd better just check and that it was right. So they invited up from London, they were in Cambridge, they invited two people up from London that were experts and knew a lot about the experimental data, and that's Rosalind Franklin and Maurice Wilkins. And Rosalind Franklin took one look at their structure and knew it was completely wrong because it was inconsistent with the data. All right, so they had to, and they realized that, so they had to eat humble pie. And it was only later when they saw this famous X-ray picture produced by Rosalind Franklin, that Watson saw this picture, that they were then able to arrive at the right model. So modeling isn't sufficient, but if you combine it appropriately with experimental work, you can you can make a lot of progress. And so the question is, could we use that same idea with understanding the development of our flour? Could we use modeling to help us interpret and understand these experimental results we were getting? But when you start to think about that, you hit a problem, because you see, Watson and Crick could model molecules with bits of paper or some rods, stiff rods, because the molecules pretty much keep their shape. But when you're dealing with something that grows, it's continually changing its shape and deforming. So what sort of material would we use to model that? What materials do we have ready to hand that grow? And the fact is, most physical materials that we're familiar with don't grow. The chairs you're sitting in don't grow. The clothes you're wearing don't grow. This lectern doesn't grow. The room doesn't grow. So there's very little, apart from biological stuff, that actually grows to a large extent. And of course, it's the biology we were trying to understand, so it's no good using that. So we're kind of stuck. But one way of getting around the problem is to say, well, let's not try and use a physical material. Let's try and model such a material in a computer. So we'd create our material within the computer, and then we could play with it, play with the rules within the computational model. Now, I'm not a computational scientist, but I was very fortunate to collaborate and interact with two very talented computer scientists, Andrew Bangham and Przemysław Pruszyńkiewicz. And both of these were fantastic people to work with, because not only did they bring the expertise in computational modeling, they also brought their own unique perspectives to the problem. And so we'd have lots of discussions, each coming from it from a different angle, and through these discussions, you gradually, certain ideas started to crystallize. And they've continued to crystallize over the years, and what I'm going to give you now is perhaps the most crystallized form where we've arrived at most recently. And the idea that's kind of emerged from all this is that behind all these shapes is the notion of conflict. Now, how does conflict help us understand shape? So let me give you some examples. So imagine we have a sheet of cells, and the top of our sheet is growing faster, that's shown in red, compared to the bottom surface of our sheet. Now, I've also marked the sheet with circles so you can see how it deforms and grows. So what's going to happen if the top of our sheet grows faster, is trying to grow faster than the bottom? Well, the sheet curls around. That's because the conflict between the upper surface that wants to grow fast and the lower surface that doesn't want to grow, by curving around, it allows the sort of compromise, a resolution of the conflict. It allows the top to grow faster than the bottom. Okay? So that's what we call a surface conflict, because it's between the two surfaces of our sheet. Here's another type of conflict. Suppose now our sheet is growing equally on both surfaces, but the center of our sheet, where it's colored red, tries to grow faster than the white region surrounding the periphery. What's going to happen now? All right, so as the center grows faster than the outside, the sheet bulges. All right, that's because, again, by bulging, it allows this conflict to be resolved. It allows the center to grow faster and the surround to keep relatively small. So it's a way, so the conflict is generating the transformation in shape. But there's a third type of conflict that so I've shown you surface and areal conflict. There's a third type of conflict which turned out also to be fundamental, but it was the trickiest one to get our head around. Because so far you'll see that those circles are remaining as circles. Everything's growing equally in all directions. But I showed you with those clones or patches on the petal that they were elongated preferentially in certain orientations. All right? So that tells you that the growth is not happening equally in all directions often. It's happening in a preferred orientation. So how do we integrate that idea? So if you're going to have an orientation of growth, you need to have something to orient it with respect to. And the easiest concept that we came up with for that was the notion that our tissue has some sort of field of arrows or orientations embedded within it, a set of orientations which we call a polarity field, represented by this field of arrows. And now we can say, suppose our tissue grows faster, every region grows faster parallel to those arrows compared to perpendicular to the arrows, what will happen? So if that happens in this case, as the tissue will grow parallel to the arrows, and you'll end up with now ellipses instead of circles, and the shape has stretched. But there's no conflict here because every bit of tissue is doing exactly the same thing. It's all trying to grow in the same way. So now let's introduce a conflict. So imagine now we have again a field of arrows, a polarity field, but now where the green region, we're going to say the things are going to try and grow parallel to the arrows, parallel to the polarity, whereas in the magenta region they're going to try and grow perpendicular or horizontally perpendicular to the arrows. All right, so there's now going to be a conflict. The green regions are going to try and grow like this. The magenta regions are going to try and grow like that. And the question is, what happens to this conflict? And there's no growth rate difference, because if you're trying to grow faster in one direction, you grow less fast in the other. So the areal growth rates here are exactly the same, all in every piece of tissue, in every region of the tissue. So what happens now? So as a result of this conflict, we get another deformation. This is a directional conflict. And you'll see now that I hope you can see that the circles are now elongated in certain directions in the green and magenta regions, showing that their growth is now being oriented. So what evidence do we have that there are these directional conflicts in the generation of something like a petal? Well, Shana Robocho, a postdoc in the lab, when she started to look at the cells in regions that form bulges in the growing snapdragon flower, what she saw was that they were elongated in different orientations. So if you look, for example, in the region indicated with the red lines, and you enlarge that region, I hope you can see the cells there. They're essentially elongated in parallel to the red line, and there's a ladder of walls perpendicular to that. That indicates that growth is being oriented parallel to the red line. Whereas if you look in the yellow region, you see the opposite. You see now that the cells are elongated in the other orientation with walls perpendicular to that. So that shows us that there's maybe a directional conflict in growth in this region that we know is going to bulge out to form part of the petal. But if there's a directional conflict of this kind, we would predict that there should be some sort of arrows or some polarity in the system that allows us to orient this growth. So then Shana went to look to see if she could find any evidence that there was a polarity, a field of arrows in our tissue. And one of the molecules that is used in plant biology to look at this type of polarity is a molecule called the pin protein, which tends to be localized at one end of the cell. And when you look at this protein in these cells, I hope you can see that it tends to be oriented always at one end of the cells, and then that allows you to draw a field of arrows on this tissue. So what that gives us then is a molecular indication that there is indeed a polarity, a field of arrows within our tissue, and this field could be providing the information that specifies whether you grow parallel, that allows you to orient your growth so you can grow faster or slower, parallel or perpendicular to those arrows. So to summarize then, we have indications that the way tissues may deform is through these three types of conflict: surface, aerial, and then the final one, this directional conflict. But then how, these transformations are relatively straightforward. Bulges, I'm generating lots and lots of domes and bulges. How do you generate something like a snapdragon flower, all right, which has a much more convoluted shape? And the solution is to combine these different conflicts, to put them together in various combinations and put them under the control of genes. And so this is how we try to do that. So now we've got a representation of a simplified snapdragon flower, a very early flower bud. And what you see is essentially a cup with these 5 lobes, which are going to form the 5 petals, lobes of the flower. And you'll see there's a set of arrows. That's going to be our field, our polarity field embedded in the tissue. It's going to give it the orientational information. And you'll also see that it's colored into different colored domains, different regions. Like there's a red region in two of the petals. That's the region that will form the dorsal part of the flower. That expresses that gene, for example, the psych gene that I showed you earlier on. So imagine the whole flower is divided into a patchwork of different regions of gene activity. And what these genes are doing is determining how fast you grow parallel or perpendicular to these arrows. That's all that's happening in this model. So you just have a patchwork of gene activities, a set of orientations, and you try and grow faster, parallel or perpendicular, and in so doing introduce these 3 different types of conflicts that I've talked about. And when you combine them together, the question is, can you generate something like a snapdragon flower? And to show you, because there's a lot of growth involved, to show you how that works, I have to shrink that to a very small size. I hope you can see it there. And then we're going to grow this tiny little thing using these principles of genes driving these conflicts. All right, so you get something that's not too far away from a snapdragon flower. Okay, so just to show you the deformations that are happening, if I rescale everything to the same size, you can see the deformations a bit more clearly. So now I'm scaling everything up so that we adjust the scale. And you can see how the tissue bends and deforms purely through these conflicts introduced by the activity of these genes. All right? So now once you have a model like that, you can play with it. You can take away some of these conflicts. What would happen, for example, if we remove the directional conflict? Let's take away all the arrows. Let's throw those away. All right, forget orientations, but we'll keep everything else the same. All right, what happens if we do that? We've got no directional conflicts. Everything's trying to grow equally in all directions now. And you don't end up with a very convincing snapdragon flower. Okay? So the directional conflicts are fundamental to generating the shape that you see. Okay. You can do other things. You take away certain genes. Let's take away the gene that's involved in the dorsal petals, the red petals in this type of illustration. So if I take away that gene, now all the petals are going to have the same information. If we run the model without those genes, then we generate this. Okay, and that's not a million miles away from the radially symmetrical mutant. So by playing with these genes and these models, we can see that these conflicts driven by genes are sufficient to generate the shapes that we observe. And so this, in a sense, is going to be our Picasso simplified bull, but now with all the features shown. And the idea is that you have a patterns of gene activity, that these set up these domains or patchwork of regions within our tissue, and then the conflicts generated by that then lead to the tissue deforming into a particular shape. Well, that's fine for a snapdragon. Can we generalize this idea? Can we look at other things, other types of structure, and see if the same principles apply? So one of the systems that we've been investigating recently is this thing. It's a carnivorous plant called Utricularia or Bladderwort. Now, this plant doesn't have roots. Okay? Instead of roots, it has these tiny little bladders that are about 1 millimeter, 2 millimeters big. It's an aquatic plant. And it has these bladders are under sort of negative pressure, and they have a mouth. And if a little crustacean or insect happens to sort of go near the mouth, it triggers the mouth and it gets sucked into the flower. Not into the flower, I mean the bladder, okay? It gets sucked into the bladder, and you can see one there that's been sucked in, and that will get digested by the little bladder and consumed and provides the plant with its nutrients. So to do this, it has a rather intricate shape. You'll see that plants produce intricate shapes when they're trying to deal with animals. So flowers do it because they're trying to deal with bees and sort of friendly visitations. In this case, there's another purpose to dealing with an animal, but the shape has the same purpose, which is to draw in an animal, but in this case, to consume. So how is this shape generated? Can we use the same understanding that we applied before? Well, we can mark these bladders, not now with pigment, but we're using this fluorescent protein, green fluorescent protein, which is shown here, which again allows us to mark the individual cells, create these patches, and look at the growth of these bladders. And if you do that and collect this information, then what Claire Bushell did in the lab was then try and come up with a model that could account for the shape and for these patterns that we see. And the model was relatively simple. It starts off with an initial shape, which is essentially a sausage shape, and we have a polarity, a field of arrows going from the stalk to the mouth, and the green stalk to the purple mouth. And then using this polarity system, we then say how fast the tissue grows, parallel or perpendicular to this polarity. And this is the model that Claire came up with. So we'll see it grow. So that's that shrunk down. Okay, so she ended up with that shape, which actually is a reasonable match to the final shape. But the question is there any way of testing whether this model is correct? Well, one of the things that it predicted was there was this field of arrows. Did we have any indication of what these arrows might be doing? Well, it turned out that we're quite fortunate because the inside the bladder is lined with a series of glands or hairs. And those glands, which you can see, well, I've enlarged some of them on the right, have an asymmetry in general. So they look like little people with arms sticking out, but the arms stick out wider than the legs held together. And so that allows you to draw a set of arrows over each one. They provide information about the polarity. And so then we can look at how that polarity is coordinated, of these glands is coordinated over our tissue. And that's shown here. So now we take our bladder and we go into the bladder and look at where all of those glands are pointing using their asymmetry. And if you do that, what you see, hopefully, is a bunch of arrows, and they're all generally pointing towards the mouth. Very similar to what we predicted based on the modeling. All right, so we hadn't anticipated this. Prior to this, we predicted this, that these arrows would be there. So that again provides some encouragement that we're on the right track in terms of understanding the generation of these shapes through these conflicts oriented by these polarities. Okay, so I mentioned earlier that the trouble with modeling something like growth is there's no material that resembles, that grows. We can't. So I had to demonstrate everything with a computer because materials tend not to grow. But there are exceptions, not to do with growth, but to do with shrinkage. And I'm rather grateful to Mark Miodownik for drawing this to my attention, which is there are certain plastics that shrink when you heat them, okay? And so what we can do now shrinkage is just the same as growth, but negative growth. You can think of shrinkage as just the opposite of growth. But everything I've told you about conflict in relation to growth applies equally well to shrinkage. So we can use shrinking plastic to illustrate those principles now kind of in reverse. And so this is where we start a demonstration. So I'm going to see if this works. So I'm going to try and shrink some plastic for you. Right, let's see. Okay, so I've got a disc of this plastic, and the outside is colored black. So that means it's going to absorb the heat faster than the rest of the plastic. So it's going to shrink faster. What I'd like you to think about is what shape will happen if you end up with a disc with the outside shrinking faster than the center. All right, so let's see what happens. Let's hope something happens. Oh, I'll tell you what, something is happening, but you're not seeing it. Okay. Let's try that again. What happened was, this thing refreshes, unfortunately, every so often. Let's try it again. Okay. So I'll watch that and make sure. Okay. All right. It forms a disc. It forms a sort of dome. Okay. The reason it's gone completely flat. Okay. Why has it gone flat? Because I heated it for too long. Because what happens? All right. This is the danger with doing this, but actually it illustrates a principle. Because what's happening, he says desperately. What's happening is this: the black will shrink faster than the uncolored stuff, but eventually the non-pigmented stuff will also shrink. And when that happens, it catches up with the black that's shrunk. All right, so that's why it eventually goes flat. So let's just do that third time. Luckily, I've got some. So now we should see it form a dome, and it forms a dome exactly like because it's like shrinking on the outside. Faster than the center is equivalent to growing in the center faster than the outside. I hope you can see they're just reverses. We have to refresh. Thank you. Thank you, team. Oh, no. Okay. Okay, so now quickly, we should see it shrink. We should see it form a dome, and then if I leave it longer, that dome will then flatten as everything shrinks. It's going, yeah. Okay, it's forming a dome. Now, if we leave it for longer, it should flatten. If you see smoke coming, don't worry. Because I was asked to fill in the risk assessment thing. So it clearly, nothing can happen. All right? Okay, so it's gone flat again. All right, but now we can do something else. Let's do this. Let's do the opposite one. Now, let's see. Let's just refresh it. Right. Okay, now I've got the opposite. Try and think what's going to happen now. Now the center is black. So what's going to happen if the center is black? Now it forms a sort of saddle. Okay. It forms a saddle, and it's going to flatten after a while. It forms a saddle because the outside doesn't shrink, the inside shrinks faster, and that conflict generates a sort of ripple or saddle on the outside. So what I hope I've shown you is how with shrinkage, by playing with shrinkage, we can actually arrive at the same principles that we could think about with growth. Now, in this case, these patterns, these different patterns of shrinking were put by me drawing carefully colouring in these different regions. But in biology, in nature, what's happening is that genes are putting those patterns in. Genes are putting the patterns that determine whether something grows or contracts. In plants, almost entirely it's to do with growth, that the genes are creating conflicts to do with growth. And so you end up with the deformations that generate the shapes, all the flowers and leaves and so forth that you see. In animals, there's a slight difference because you also have contractions. So animals can shrink effectively, or bits of them can shrink. So with animals you have shrinkage as well as growth, but essentially the same principles apply. You end up with these conflicts that can account for the various tissues that we see. Now once you have this idea of conflicts, you can then start to do things. You can then create conflicts at will. You can start to play sort of and say, what sort of shapes could we produce that maybe nature has never produced, but we can use these principles. So So this is an example of that. This was a case where we had a cup shape. We put a polarity field on it, and we just thought, well, let's throw a few different regions growing at different rates. What shape might we get? All right, and this was the shape that we generated. So we weren't aiming to produce a flower here. We were just aiming to just play, and we produced a shape, a rather attractive shape, to my mind at any rate. It looks organic because it's been generated by these organic principles, but it doesn't resemble anything. Somebody said it looks like garlic bulb or something like that, but that's not what we were trying to do. So anyway, I showed this to Rob Kessler, who's professor of ceramics at Central St. Martin's, and he took this shape and showed it to a glassblower. And the glassblower, hold on. Yeah, this is the shape the glassblower produced, inspired by that. And I've got it here, okay? So again, I think that's rather nice. Okay. But it's interesting because normally an artist would be inspired by a natural form, a natural flower, or a natural leaf, or a natural shape. In this case, they were inspired by something that doesn't exist in nature, but that had been produced by sort of natural principles, the principles of growth and conflict that I've described. Okay, I've talked a lot about conflict, and I was talking to a colleague of mine saying how, isn't it amazing, all the shapes that you see in a garden, they're all to do with conflict, isn't that incredible? And they said to me, why are you so negative? Why do you talk about conflict all the time? Can't you get a bit more positive about life and see, the good things instead of harping on conflict? Why are you so neggy? That's what one of my sons would say. And so I thought about this, and it's true that conflict often produces negative outcomes. Okay, so that's of course the case, but that's not always the case. And there are cases in which it has very positive outcomes. And the classic scholar, Greek scholar, Edith Hamilton, in her book, The Greek Way, she says, "Great art is the expression of a solution of the conflict between the demands of the world without and that within." And you can see some of those conflicts, actually, in Picasso's bulls. There was a conflict between simplicity and complexity. There's also the conflict in many of Picasso's drawings between a three-dimensional object and trying to represent it in 2 dimensions. That's another conflict. But conflicts are also found in science. If you think about the way science proceeds, you have maybe an observation, you come up with a hypothesis, you then do further experiments, you show that the hypothesis doesn't fit the results, so that you resolve the conflict by coming up with a better hypothesis. You then go and do more experiments, you find they still don't fit exactly, so you then come up with a better hypothesis. And so science really depends on continual resolution of these conflicts between hypothesis and observation. If you took that away from science, it wouldn't be what we recognize today as science. So conflicts, but in both science and art, are actually a source of inspiration and creativity. They're a driving force for creativity. And it's in that sense that I've been using the term today. Because in evolution, it's as if living systems stumbled on a remarkable way to create tissues and shapes through genes introducing conflicts into these tissues. And through that process, we end up with all the wonderful shapes that we appreciate and that surround us, including the ones that continues to fascinate us and intrigue us. Okay? And on that note, I'd like to finish, but I would like to thank some of the people involved in what I've been describing. Research depends on working with a great team of people, and I've been very fortunate over the years to work with some fantastic people that have really, it's just been a joy to work with. This is the group photographed quite recently on the Suffolk coast. And I particularly want to mention some of the people that contributed to the work that I mentioned today. I'd particularly like to mention Richard Kennaway. Richard wrote the software that I showed that I've been using to model the tissue growth. And without his contribution, pretty much nothing, a lot of the things I said just wouldn't have been possible. So I'm incredibly indebted to Richard for doing that. And he continues to help us in that way. I'd also like to thank Shana Rubocho. I mentioned she was critical in unraveling the conflicts behind the generation of the snapdragon shape. Okay, so I'm very grateful to Shana for that very insightful work. I'd also like to thank Lucy Copsey, who does a lot of the breeding and crossing of the antirrhinums. Paul Southam, who does a lot of the modelling as well, and image analysis that I described. And also I'd like to thank the Bladderwort team, Karen Lee, who produces these fantastic 3D images and initiated a lot of the Bladderwork work. Claire Bushell, who I mentioned, did the modelling that I described, and Chris Whitewoods and Min Long Kui. Min Long's in China, so you can't quite see him there, but they worked on generating those transgenic plants with the GFP sectors. I'd also like to thank Rob Kessler and James Maskery for the glass work that I showed you, Jonathan Keep for the pottery. I'd like to thank Katrina Ross for organising this event. And finally, last but not least, I'd like to thank my family, many of whom are here, for all their support over the years, and particularly my wife, Lucinda, who most recently has had to suffer volumes of shrinking plastic in the kitchen. So thank you very much.
So, are there any questions for Enrico? Yeah.
Make an observation, it's like beating metal. It's exactly what you do when you blacksmith.
What, sorry?
The differential shaping. Yeah. The differential shaping is exactly what you do when you go do blacksmithing or beat metal.
Well, that's very interesting. Do you do that?
I have done. I'm a bit old now.
Very interesting. Thank you.
Yes, in the middle there. Thank you.
Hi, thank you for your lecture. It was really interesting. I was just wondering, when you showed us originally the ideas of the different types of conflict, and you started off with a square with the circles on, and it looked as though when the squares deformed, the circles did too. And I was wondering, if you didn't start with a square, if you started with like a circle or an oval, would the circles on the surface also deform in the same way?
Thank you. Yeah, it doesn't. The tissue would still curl around itself, and the circles would deform in a similar way as well. Yes. So, but the actual shape you would generate would be different because of your starting shape. So instead of having the sort of edges coming down, as you saw, there would be a smoother outline perimeter to it. But essentially, you'd expect the same type of transformation, irrespective. The curvature would be very similar, and the circles within would deform locally in a very similar way. Yeah. We have done that, by the way.
Yes, in the middle again, please.
Hi, thanks for your talk. So I understand how a plant can generate pattern in the up-down axis and also radially, but I don't understand how they can generate pattern with a dorsal-ventral axis. Like, how does antirrhinum decide which side of the flower is going to be dorsal and which side is going to be ventral?
Okay, that's another good question. So the upper part of the flower actually is positioned in relation to the growing tip of the plant. So if you look at a snapdragon, you'll see that the flowers come off the side of an axis. There's a main axis, and the flowers come off the side of that axis. And so if you were to look at where the buds form, they don't actually form the top of the growing tip, they form to one side. And that means they've got a tip above them. And we think that there's information coming from that asymmetry. That is, that they're referencing the structure that's above them, which is the growing tip. So it's not gravity or anything like that, because if you turn the plant upside down, they will still orient in the same way in relation to the tip. But we think it's related to how the genes are being switched on in relation to the tip of the plant.
There was one here. Is that right? Yeah.
Could you say exactly what the nature of the polarity is, the molecular nature of it?
Right. What's the molecular nature of the polarity? So I showed you those pin proteins. And what those are doing is they're transporting a molecule called auxin, which is a plant growth hormone. And the idea is that the transport of the auxin is somehow involved in coordinating between the cells the polarity, the orientations of these asymmetries within the cells. One question is how these asymmetries are generated within the cells. And there's a lot of debate a hot topic in plant biology as to how these polarities are being generated. There are various different models. Some, for example, postulate that cells have sort of intrinsic system that can polarize molecules at different ends of the cell. Some postulate that they sense the direction in which this molecule is flowing, this auxin molecule. So it's not known. That's the short answer to your question. There's a lot of ideas as to how these proteins are being asymmetrically localized, but they're thought to be coordinated through this auxin molecule.
I was wondering what was special about the number 5.
What's special about the number? Would you like to elaborate?
Well, it seems to me that, am I right in thinking these have 5-fold symmetry? Is that
right?
Is that right? I mean, in some plants like crucifers, I guess, have 4-fold. You know, what's?
Yeah. No, you're right. So
there must be some simple little rule there.
Yeah. So why 5? So you can come up with theories. For example,
I think it would be 6 or.
You would, maybe. I don't know
At least
why
I
you'd
would.
think it would be 6. Why would it be any number?
Why would it be any number?
So we can get, so people, when they see numbers, they can get, people get very exercised about this. 5, for example, is a member of the Fibonacci series. All right, so people say, oh, 5 it's very significant because it's a member of and you have 5 petals, and 3 is another member of the Fibonacci series. But actually, some of our mutants with radial symmetry, in fact, I think the one I showed you actually has 6 petals. Okay, so it's not, I don't think there's anything magical about 5. I think that it so happens that in this family of the snapdragons, they tend to have 5 petals. What's curious is that you conserve that number. So you're quite right. If you look in things like the crucifery, they have 4 petals. So it's not that 5 is intrinsically better than 4, but maybe once you've arrived at a particular number, then you want to you basically organize your developmental system according to that number. And so it becomes quite difficult then to change your number. That's very common in a number of situations. Once something becomes sort of locked in, then you become punished, because then you have to change a number of things as well as changing that number. So in the Snapdragon family, 5 seems to be the number that's been locked in. But I don't think there's anything magical about 5 in itself. There are people that do believe, I think the quincux theory or something like that, there are people that think that there are these very special numbers that have a major significance, but I don't agree with that.
I wondered if the mutant plants still got pollinated, and what attracted them, or do they not need attraction?
Right.
Was it by the bees, or?
Do you mean the radially symmetrical flower?
Well, the chaps who didn't have any red anywhere near them.
Right.
Yeah.
So you mean the koi one?
Yes.
Right. Does it get pollinated?
Yeah.
It only gets pollinated by geneticists.
Oh.
Okay. But bees, I've never seen a bee go towards one of those things.
Right.
So it wouldn't exist in nature. It would be a dead loss.
Right.
That you don't find them naturally.
I just wondered if they would be selected when we got rid of the bees.
Would they be selected when you... Well, are we going to get rid of the bees? That's news. But I know what you mean. Would they then do well? Well, the trouble is nothing will do terribly well.
Right.
So, at least, well, the only things that will do okay will be wind-pollinated plants if we didn't have bees. Right. So actually everything would be doomed. All of the snapdragons, whether you had...
Are there no other insects around that will step in?
Oh, I see. You mean if there were other insects?
Yeah.
Yes, if there were other insects, maybe they would. But again, there's the question, would those, like beetles, for example, would those beetles... So snapdragons themselves, the normal one, would go because that does require bumblebees. The mutant that I showed you might not, might get pollinated by beetles, but the problem is it doesn't produce any male organs, okay? So you saw that tube coming out of the center, it's female structures. So actually it would have no pollen to pollinate itself. So either way you look at it, it's doomed. It's doomed.
Are there any more questions? Yes, please do in the middle.
Hi, so you mentioned that in animals, you don't really see much shrinkage, but more growth. No, sorry, that in animals you do see shrinkage and growth, but not in plants. So I was wondering if you had ever observed that in plants, or if you were trying to sort of, for this computational modeling, include further variables such as cells not growing or cells dying, growing slower than the medium. So yeah, essentially what the next steps for this modeling would be, or...
No, I think that's a good question. I think By the way, when people say that's a good question, they usually mean that's a question I've already thought about. But actually, a lot of these questions are really good, because I hadn't thought about them. So, so yeah, what was the question? Yeah, contraction. Yes, you would use that particularly to model animal development. So everything I've said to you about modeling plant development, we can introduce negative growth, we can introduce contraction into our modeling system, exactly as you say, and you can generate deformations. You can, for example, simulate a process called gastrulation, where the animal tissue forms a furrow through differential contraction on one surface. And so you can build on what I've shown you to actually model animal tissue development as well. So it's a very general framework, and you absolutely could incorporate, you just put, instead of a plus, you put a minus, and you'd use exactly the same system.
There was one there, yes.
Thank you for a very, very interesting talk. Now, I'm wondering about all the complexity in the shapes of flowers, and at what stage will the cellulose wall be laid down that will bring it to its final shape, and what actually would cause the final shape to be established?
Do you mean how is what I've said related to things like the cell walls and the structures of the cell walls?
Yes.
Yeah, that's another great question. So I have thought a bit about that one. So yeah, when we're modeling growth here, we're just been saying grow more in one direction versus another direction, for example. But what actually causes growth to happen? And plant growth is driven by turgor pressure, largely. The cells are under pressure, and whether they grow in one direction or another depends on how stiff the walls are in each direction, which is determined by these cellulose fibers. All right. So when we try and think about how do genes actually influence whether something grows more in one direction or another, or the extent to which it grows, that's going to, we think, come down to how do you lay down your cellulose fibers, how do you reinforce them, how do you bond them together. And so all the shapes at that level will come down to cellulose fibers. So in a sense, or other types of fibers in the cell walls. So, but it's kind of like what you're doing is you're zooming in to the question of what is driving growth and growth orientation. In a way, I gave a slightly zoomed out version and saying the consequence of these cellulose fibers being strengthened in some orientations versus others would be that you would generate conflicts at the tissue level that would then cause the deformations that you see. So it's
something to do with shrinkage in animal cells, which don't have cellulose and are therefore easily susceptible to shrinkage, whereas plants, because they have the cellulose, won't do so.
Right. So, how is this related to animal development? So, in animals, you're right. You don't have the cell walls. The shape of the cells is more determined in things like the cytoskeleton, the actin and myosin, which are these contractile filaments within the cell. And it's things like that that will then determine whether you contract more or you're more rigid in certain orientations than others. So the conflicts are going to be produced by different types of molecules, by a different mechanism. But the nature of the conflicts is going to be very similar, in the sense that you'll end up with surface or aerial or directional conflicts, but the molecules doing it and the way they're being done in animals is different from the way it would be done in plants.
Okay, well, let me thank you for your questions. And let me join with the last questioner in saying thank you, Enrico, for such a wonderful lecture.
Thank you.
Thank you.
Thank you.